Introduction. Glaucoma is a group of eye diseases that damage the optic nerve and can lead to vision loss if not treated. High intraocular pressure, IOP, the fluid pressure inside the eye, is a major risk factor for glaucoma. Standard treatments like eye drops and surgery focus on lowering IOP, but growing research suggests that diet and nutrition may influence glaucoma risk and progression. For example, diets rich in vegetables, sources of nitric oxide nitrates, have been linked to lower glaucoma risk. Personalized nutrition, or precision nutrition, is the idea of tailoring a person's diet to their unique biology, including their genes and metabolism. The new field of nutrigenomics studies how genetic differences affect the way our bodies process nutrients, like fats and carbohydrates, and how these interactions impact health. In glaucoma, nutrigenomics could one day help us recommend the best balance of fats, carbohydrates, and proteins for each patient based on their genes. This article explores how key genes involved in fat and carbohydrate metabolism, notably APOE, PPAR family genes, fats, and NOS3, might guide personalized diets for glaucoma, how clinical trials could test such approaches, and what ethical and practical issues arise. Genes and macronutrient metabolism. Certain genes play major roles in determining how our bodies handle fats and carbohydrates. Variants, different versions, of these genes can change metabolic pathways. In the context of glaucoma, several genes are of interest. APOE, apolipoprotein E. This gene makes a protein that transports cholesterol and fats in the body, especially in the brain and retina. There are 3 common APOE variants, called EP2, F3, F4. People with the F4 version tend to have higher blood cholesterol levels. In general nutrition science, APOE4 carriers often show larger cholesterol changes when they change their intake of saturated fats. For example, cutting saturated fat often lowers cholesterol more in APOE4 individuals than in others. In glaucoma research, some studies even suggest APOE4 might protect the optic nerve from damage, though the picture is complex. From a diet viewpoint, an APOE4 carrier might benefit especially from a low saturated fat diet and increased healthy fats, in line with heart-healthy guidelines. PRs, peroxisome proliferator activated receptors. These genes, especially PPAR and PPAR-GONG, are regulators that turn on or off pathways controlling fat and sugar metabolism. The PPAR-GONG gene has a well-studied variant called Pro12-ALA. People carrying the ALA12 variant often have greater sensitivity to different types of fat in the diet. For instance, one trial found that carriers of PPAR-GONG ALA12 lowered their cholesterol and triglyceride levels more when their diet had a higher ratio of unsaturated fats, polyunsaturated, saturated fat. Another study showed that ALA12 carriers lost more weight on a Mediterranean-style diet rich in olive oil, a monounsaturated fat, than on a standard low-fat diet. In short, PPAR variants influence how well someone responds to healthy, unsaturated versus less healthy fats. For glaucoma patients with these PPAR variants, emphasizing omega-3 and monounsaturated fats from fish, nuts, and olive oil over saturated fats may be particularly beneficial. FATs, fatty acid desaturase, the FADS genes, FAS1 and FAT-S2, control how our bodies convert short-chain fatty acids from plants into the long-chain omega-3 and omega-6 fats that we need. Variants in fats strongly influence blood levels of omega-3 fats like EPA and DHA. A recent review of many studies found that certain FATS1 single-letter changes, like RSM14537, are consistently linked to lower blood EPA-DHA levels. In other words, people with those FADS variants convert plant omega-3s like ALA and flaxseed into the active forms, EPA-DHA, less efficiently. For eye health and general health, omega-3s are important. If a glaucoma patient has a FADS variant that limits their omega-3 production, they may need to eat more direct sources of EPA-DHA, such as fatty fish or algae oil supplements, to compensate. Tailoring the balance of omega-6 to omega-3 fats based on fat's genotype is a key gene-diet interaction to test. NOS3, endothelial nitric oxide synthase. This gene makes an enzyme that produces nitric oxide, NO, a molecule that relaxes blood vessels and promotes blood flow. Good blood flow is important for the optic nerve. Certain variants in NOS3, like GLU298-ASP, affect how much nitric oxide a person naturally produces. Diet can boost nitric oxide too. For example, dietary nitrates, found in beetroot, spinach, and other green vegetables, are converted into nitric oxide in the body. Notably, a large population study in the Netherlands found that people with higher nitrate intakes had a significantly lower risk of developing open-angle glaucoma, even after adjusting for eye pressure. This suggests nitrates NO help protect the optic nerve in ways not captured by pressure alone. Thus, a patient with a NOS3 variant that lowers NO production might benefit more from a nitrate-rich diet, lots of leafy greens, beets, etc., or other no-boosting nutrients, like arginine from nuts and seeds. Each of these genes illustrates a potential gene-macronutrient interaction. APOE links to cholesterol and fat, PPARs link to types of fat and sugars, FADEs to omega-3 availability, and NOS3 to vascular health. In practice, one framework could be to genotype patients for these key variants and assign them to broad diet patterns. For example, an algorithm might score each person on an APOE profile, or FADS profile, and then recommend a diet higher or lower in certain fats accordingly. In research settings, scientists could also use multi-gene risk scores or decision tree algorithms that incorporate several variants at once. See personalized nutrition study below. Designing adaptive diet trials in glaucoma. To test these ideas scientifically, we would need clinical trials designed for personalized nutrition. Traditional trials, where everyone in a group gets the same diet, may not catch individual effects. Instead, trials could be adaptive and genotype-informed. N of 1 individualized trials. In an N of 1 trial, each participant acts as their own control. For example, one design might have a glaucoma patient follow diet A, e.g. higher fat, lower carb, for several weeks, then switch to diet B, lower fat, higher carb, for several weeks, possibly with a washout period in between. During each period, we would record outcomes like IOP, visual field tests, and blood biomarkers. This way, each person can discover which diet works better for them individually. Such designs have been used in metabolism research. The Westlake trial, WMAC-Nutter, is a good example. Researchers had healthy adults rotate between a low-fat, high-carb, and a high-fat, low-carb diet, while continuously monitoring their blood glucose response. They used a Bayesian model to predict who responded better to each diet. A similar approach in glaucoma could use continuous IOP monitors. There are now contact lenses that can track pressure, or at least frequent eye exams, along with blood metabolomics, to see which diet period led to better ocular outcomes. Randomized adaptive trials. Alternatively, one could run a multi-arm trial where groups are stratified by genotype. For example, participants might first be genotyped for APOE, PPAR, FATs, and NOS3 variants. Then each person is randomized to one of several diet plans, e.g. a high omega-3 diet versus a standard diet versus a high-protein diet. After an interim period, the data can be analyzed and the trial adapts. People who are not improving might be crossed over to a different diet, or new participants might be assigned based on lessons learned so far. This could be done with Bayesian adaptive design methods. The key point is that assignment can change based on emerging results to maximize each person's benefit. Multi-omics phenotyping. In all these designs, the trial would integrate genomic data with metabolomic data, profiles of small molecules in blood or urine, and ocular phenotypes, IOP, and visual field. For instance, researchers might measure a panel of blood metabolites like lipids, amino acids, nitric oxide markers before and after each diet phase. These metabolomic fingerprints show how the body is responding at a biochemical level. In fact, a recent personalized nutrition trial classified people into metabotypes using 4 blood markers: triglycerides, HDL cholesterol, total cholesterol, and glucose, and then delivered diet advice tailored to each metabotype. After 12 weeks, this personalized approach significantly improved diet quality and reduced cholesterol and triglycerides compared to standard advice, for example, and LDL levels were lowered significantly. This shows how metabolomic profiling can guide and verify personalized diet effects. In glaucoma trials, we would do the same, use metabolomics to adapt the diet and also to see if beneficial changes in metabolism correlate with improvements in IOP or visual field. Ocular outcomes. The main outcomes in such trials would include IOP measurements and visual field tests. IOP is usually measured in the clinic, e.g. with a tonometer, and reflects pressure control. Visual field testing checks peripheral vision and is a standard way to gauge glaucoma damage. Ideally, trials would measure both IOP and visual fields repeatedly. For example, after each diet period, an eye doctor could perform a visual field exam to see if any slowdown in vision loss occurs. If a particular diet consistently leads to lower IOP or less worsening of visual fields in certain genetic groups, that would be strong evidence of a beneficial gene-diet interaction. By using adaptive designs and modern technology, wearables, and digital diet logs, these trials could learn quickly which diet patterns work for which genetic profiles. The Food for Me study, an EU-wide personalized nutrition trial, showed that telling people their gene results did lead to healthy changes, and the Points weight loss trial used genotyping to define fat responders versus carb responders groups. We can apply similar ideas in glaucoma. For example, in the Points trial, subjects genotyped as carbohydrate responders or fat responders were randomized to matching diets, but results showed no large weight loss difference between genotype concordant and discordant diets. This highlights a challenge. Even if genes suggest a diet, the real-world effect may be small or hard to detect. Careful trial design with enough participants and good outcome measures is crucial. Ethical, privacy, and practical challenges. Personalized nutrition carries ethical and privacy issues. First, the scientific community urges caution. As Bergman et al. Note, until the scientific evidence concerning diet-gene interactions is much more robust, the provision of personalized dietary advice on the basis of specific genotype remains questionable. In other words, telling a patient, "Eat this way because of your gene variant," should be done carefully so as not to promise more than we know we can deliver. Patients must give informed consent and understand that such diets are experimental and supplemental. It's also vital to remind patients never to stop proven glaucoma treatments, eye drops, etc. Diet advice can complement treatment, but not substitute it. In fact, recent reviews of diet and glaucoma emphasize lifestyle measures: healthy weight, fruits, vegetables, moderate caffeine. In addition to conventional therapy, genetic data privacy is another concern. DNA information is highly personal. Patients need assurance that their genotype and metabolomic data will be kept secure and used only for their care or authorized research. Laws like the Genetic Information Non-Discrimination Act in the U.S. and similar regulations elsewhere must be followed to prevent misuse by insurers or employers. Databases of nutrigenomic results should be de-identified and protected. Finally, translating this into clinics is challenging. Many doctors and dietitians currently lack genetics training or easy ways to interpret gene reports. Personalized diets can be costly: genetic tests, repeated metabolomic labs. We must also consider equity. If only wealthier patients get genotype diets, that could widen health gaps. All these issues, scientific uncertainty, consent, privacy, cost, and fairness, must be addressed. Work by Bergman et al. And others lays out these bioethical considerations for nutrigenomics. Open communication, transparency about benefits/limits, and clear guidelines will be needed as the science develops. Priority gene-diet interactions for validation. Based on current knowledge, the following gene-diet pairs are top priorities for study in glaucoma. APOE variants, saturated versus unsaturated fats. APOE influences cholesterol transport. People with the F4 variant often have higher cholesterol and show strong responses to saturated fat intake. Clinically, it will be important to test if APOE4 carriers with glaucoma do better on diets low in saturated fat and higher in healthy unsaturated fats, nuts, fish, olive oil. PPAR Gang, Pro-12-ALA unsaturated fats. The PPAR Gang ALA-12 variant has shown stronger improvements in lipid levels when diet includes more polyunsaturated monounsaturated fat. For example, ALA-12 carriers lost more weight on an olive oil-rich diet. Trials should check if glaucoma patients with this PPAR variant experience better eye pressure control or neuroprotection when on a Mediterranean-type diet versus a standard low-fat diet. FATS1 RS74537 and related omega-3 intake. Variants in the FATS genes greatly affect how much EPA-DHA, long-chain omega-3s, enter the blood. Individuals with low converter FATS variants likely need extra dietary omega-3. It's a priority to see if glaucoma patients with these FADS variants benefit more from increased consumption of fish or algae oil supplements versus patients without the variant. NOS3, e.g., Glu-298-ACE-P, dietary nitrates. Given the Rotterdam and Nurses Health Study findings that nitrate-rich diets, beetroot, leafy greens, are linked to lower glaucoma incidence, it would be valuable to validate whether NOS3 gene variants modify this benefit. For instance, people with a less active NOS3 form might see greater IOP lowering or optic nerve protection from a high nitrate diet, while others might not. Other interactions are possible, e.g., genes affecting carbohydrate tolerance might guide glycemic index of the diet, or inflammation-related genes with calorie intake. But APOE, PPARs, fats, and NOS3 are strongly backed by metabolism science. These hypotheses can be tested in carefully designed trials. For example, one could recruit two groups of glaucoma patients, with and without a given gene variant, put them on diets that differ in the nutrient of interest, and measure IOP and nerve function over time. Successful validation would mean identifying which diet helps which genetic subgroup. Conclusion: the idea of personalized nutrition in glaucoma is still emerging, but it promises a more tailored approach to eye health. By studying how genes like APOE, PPAR guang, FATS1, and NOS3 interact with fats and other nutrients, researchers hope to figure out if certain glaucoma patients can benefit from specific macronutrient changes. New clinical trial designs like N-of-1 studies and genotype stratified adaptive trials can test these diet gene strategies effectively. However, this field faces hurdles. The evidence linking diet to glaucoma is mostly observational so far, and ethical issues like data privacy and equitable access must be handled carefully. For now, diet advice for glaucoma remains general. Keep a healthy weight, eat plenty of fruits and vegetables, and follow medical treatments. But as science advances, we may one day supplement that advice with genome-guided diet plans. Until then, research must proceed with rigor and care to ensure patients truly benefit from any nutrigenomic guidance. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.